Pablo, I have two questions. The first is if you know if MIR190 is regulated at the transcriptional level, and who is the regulator? And the second question is how Fatiga regulates CIMA?
It again, please,
Well,
Mario.
The first question is if MIR190
yes.
is regulated by hypoxia, increasing its transcript levels.
Yes.
And the second one is how Fatiga regulates CIMA, if you know.
How Fatiga regulates CIMA.
Yeah.
Well, Fatiga mediates hydroxylation of CIMA in one key prolyl residue. This enables the rapid degradation of at the proteasome.
Okay.
Regarding the induction of MIR190, the answer is yes. It's induced by hypoxia at the transcriptional level. And surprisingly, this is independent
of CIMA.
On CIMA. So there is not a loop that closes on itself, limiting the activation of the system.
And do you not have any candidate?
How it's regulated? Well, there are other transcription factors that are known to be regulated by hypoxia as well as, for instance, NF-kappa B, but we haven't done anything to look for the regulator of this microRNA.
Thank you. Rafael, please.
Pablo, very nice talk. Thank you. Just to hear your opinion, because the concept of hypoxia in terms of experimental design is complicated, because we many times culture cells under conditions that would be called hyperoxia, right? Because in a cell culture room, we are under 21% oxygen, and actually in the tissues, we see maybe 3 to 5, 6% oxygen in humans. So the questions are, under which oxygen concentration the HIF system starts to operate in micromolar level, and also how much does the Drosophila oxygen sensing translate to human cells or human tissues?
I will start by the last one. The system is highly conserved at the cellular level. We have the transcription factor, the prolyl hydroxylase, everything seems to be quite conserved. Regarding if hypoxia is really hypoxia or not, we work in vivo. So our studies are carried out in flies. So that's easy. I mean, it's 5% oxygen. No doubt that this is below atmospheric normal oxygen, so we do so. And operatively, when we move to cell culture, the system is activated at around 1% oxygen and below that. If we go down to anoxia, then it's blocked and it doesn't work any longer.
Any further questions? Yes.
I think that the problem with that is that it's very, very relative. And we work with cells all the time which are kept at 21% oxygen. And then we draw conclusions. And when you go to experimental designs, every single laboratory calls hypoxia one thing or the other. And there is a need, I think, for defining what are we talking about. In our laboratory, below 3% oxygen starts becoming a hypoxic response. But above that, we can call it low oxygen concentration, but it's not really hypoxia.
Yes, I agree. Our advantage is that we work in vivo, so it's quite physiological. The concentration that we use could be compared to the summit of the Everest, something like that. That's about 5% oxygen.
Yes, Eddie?
Tumors in humans are supposed to have a hypoxic medium, or environment, yeah. So how does this connect to human cancer, perhaps?
I would say that the conclusions that we get from the fly system could in principle be applied. Of course, we have to prove that, but it has been done in experimental tumors in Drosophila. The HIF system is also activated. This is not published, but has been done. I saw it in some meetings.
Pablo, very nice talk. I was wondering, what is the physiologic relevance of CIMA target genes? Are they related to angiogenesis, inflammation? And the second question is whether any HIF-independent pathway is relevant there, like NF-kappa B, for example.
There are many target genes in mammalian systems. As you know, there are about 700 to 800 target genes. In flies, it's a little bit less, could be 100 or 2. And we have a little bit of everything. We have systemic response genes, cell autonomous important genes. The systemic genes are, for instance, the ones that control the outgrowth of the tracheal system, which is a process analogous to mammalian angiogenesis. Genes that control the growth of the whole body of the fly, as well as individual organs. And the ones that work at the cell level are mostly metabolic. There is a general tendency to block mitochondrial oxygen-dependent metabolism and to favor anaerobic fermentation, just dependent on glycolysis.
Okay, thank you so much again, Professor Warner.